So yeah, thank you very much for giving me the opportunity to talk about some of the lab's research today. It's been a great meeting so far. And sort of when I was discussing, why do you want somebody who deals with insulin secretion and really is quite scared to go outside of the pancreas for this meeting? And Caroline said, well, of course, because insulin resistance, insulin secretion is a bit of a futile cycle and they both go hand in hand. So I think it's important to know about both things. And I should apologize, I'm a bit out of breath because inspired by Andre Carpentier's talk and others yesterday, I've been to have a heavy session in the gym to try and reduce my levels of ceramides. So I'm going to speak to you today about islet heterogeneity. When I say islet heterogeneity, I'm really going to focus on beta cell heterogeneity. I'm also going to focus on heterogeneity in GLP-1 receptor expression and signaling and how this impacts upon hormone release. But before I get started, you'll be aware that the UK or the United Kingdom or Great Britain or whatever we call it, it's probably soon to be England when we dismantle the Union, is not the most popular country in the world. We've given rise to an entirely new variant of COVID, which resulted in a second lockdown. We're incubating another variant of COVID as we speak, and we left Europe so we could build our borders back up. So that's made it quite hard to attract good people to come and work in the UK. And it's always been hard in Birmingham because it has a reputation as being fairly industrial. But I want to show you why you might want to consider coming here as a tourist, and also why you might want to actually come here to work with us. And that's because we've got some of the best urban planning in the world, stretching back to the 1970s. This is a walkway through the centre of town that you probably don't want to go on a dark night. Our architecture is stunning, literally stunning. So it's in a constant sort of state of disrepair, being kind of ripped down and built back up. But we are of the Victorian era, slightly balanced with some 70s monstrosities after we got razed to the ground during World War II. Nightlife is tremendous. So this is Lab Christmas party here. This is me dressed up for a night out, and Broad Street is where it all happens, and we do like to party here. And the transport links are fantastic. You can probably spend about 3 hours of your life on this motorway, which is called Spaghetti Junction, and it's the confluence of the motorways from the north, south, east, and west. And it means once you get off Spaghetti Junction, you can pretty much get to anywhere quickly in the country because we are in the Midlands. But in all seriousness, the best reason to come here is because we are the home of heavy metal, Judas Priest, Led Zeppelin, and of course, Black Sabbath. And in fact, Ozzy's son, Louis, was my old neighbor up in town. So with that said, what I'm going to talk to you about today is really heterogeneity across the islet, from the single beta cell through to the single molecule on the beta cell itself. And suffice to say, over the last 10 years, there's been a real explosion in our knowledge and understanding about diversity between beta cells. And this isn't really new. We've known for about 50 years now, several studies by Paulo Maida, Marcia Herriat, and others, that beta cells are quite heterogeneous. And it's only been in the last 10 years we've got a really good handle on this with the advent of single cell screening technologies. So ubiquitous single cell RNA-seq and CyTOF and so forth. We can now sort of broadly characterize beta cells according to their activity patterns, so pace setters or followers, using ontogenetics and modeling, their insulin granularity, so high, medium, and low, based upon flow cytometry, their expression or not of insulin gene and protein. So we have cells which have high and low insulin, we have extreme insulin containing cells, and we have cells that have high levels of ER stress, so UPR response. And this is observed using genetics, single molecule fish, RNA-seq, and so forth. Cell markers don't delineate cells according to functional subpopulations because the marker doesn't actually do much itself, but it allows you to segregate them and then assess how they do contribute to function. And we have cells that are UCM3 negative, positive, so mature or immature, flat top negative and positive, so proliferative and not proliferative, and RBP4 and 4+, which are highly exocytotic or not. And we can even group beta cells into protein clusters, so C1, C2, C3. And this really gives an idea of the complexity of the landscape. And a lot of people say, you know, it's almost too complex, but the reality is that, yeah, that's how organs are constructed, right? You don't just bolt together cells all of the same type and then expect them to make a gain of function. I mean, we have plasticity, we have redundancy, and so forth. About 5 years ago now, we used optogenetics to kind of look at cells which we thought were exerting some disproportionate control over the islet. And we termed these cells hubs at the time. And if you silence these cells, you really dampen down responses across the rest of the islet to glucose. And subsequently, a number of other groups showed that kind of similar cells exist, so leaders and first responders and child-rhodopsin 2 cells and even switch cells these days. But we were pretty conscious that one of the unifying features of most of these studies is these cells do have different properties to the other cells. So when we took a look at our cells here, the hubs, we saw that they had low levels of PDX1 and NKX6.1, which are beta cell maturity markers, and low levels of insulin, much lower than the rest of the population or the average. But they were energetically adapted. They were able to generate quite high levels of ATP and tolerate quite high levels of calcium. And we were conscious that these were quite biased studies. We were conscious that, you know, we'd named a subpopulation, which in hindsight, I don't think was a great thing to do. But we're also conscious that these cells are probably the tip of the iceberg and there's a continuous distribution of these cells in the islet. And one of the things that was kind of driving me is that if you look at an islet, you'll see cells that are mature and you'll see cells that are less mature. So they've been through differentiation, but they'll have lower levels of, for example, transcription factors like PDX1, MAFA, and NKX6.1. And it makes no sense to me why those cells would have come about in an evolutionary sense if we then just had to build in mechanisms to kind of circumvent the functional deficiencies, because of course these cells release low levels of insulin and express low levels of insulin. And I actually thought, well, you know, if we just go back to basics, maybe actually these cells are doing a little bit more than we think we are. And maybe these cells are actually important in the landscape of the islet in order to sort of engage in plasticity and high secretion responses, but as well to avoid over stress on the islet. So with that in mind, we hypothesized that heterogeneity across beta cells is important. And if we get rid of the immature cells, actually, counterintuitively, we should see the islet begin to fail. So what we did is we came up with a model where we had our normal islet here, which is B-norm islets. This is all work done by Daniela Nasteska, a talented postdoc in the lab. And these islets were normal, and you can see that we have cells with high and low PDX1, and we just kind of arbitrarily define them based upon this. Same with MAFA. And then what we did is we added back in transcription factors, so PDX1, MAFA, and we were able to drive cells that were low expressors to this kind of high bracket. So now all the beta cells were unanimously mature. So a good analogy is sort of like if we took society, right? And we decided just let's get rid of everybody apart from people my parents' age. You know, my parents would think this is a fantastic thing to do. They're right about everything, and their generation, you know, has been right about everything going forwards and always have a lot of wisdom. But the reality is that I don't think society would function very well if just my parents' generation were present. We need people from all age groups in order to have a healthy, robust, and plastic society. So this is what we did here. We went from our sort of united, sort of heterogeneous society to our kind of more elderly society, let's say. We knew that we were targeting these cells because we can take islets where we have BFP1 as a fusion construct of PDX1. So you express BFP as a function of PDX1 expression. And you'll see that cells that were historically low in PDX1 now become high in PDX1. This is protein, this is the gene. So we're confident that you're switching cells from a PDX1 MAFA low phenotype to PDX1 MAFA high phenotype, whereas you're not really doing anything with the PDX1 MAFA high cells. And actually, this sort of opened up a more interesting question: is when you're using viral approaches to perform these manipulations, it turns out that the viruses target the less mature cells, which I think people like Decio Azira had sort of shown previously, but really came to the fore here and was actually a major advantage. able to assign some causality to this. This is an optogenetic transcription factor construct system, which is PDX1 with an neon green fusion. And when you shine blue light, the C120 fuses with this and drives expression, and you're able to now essentially mature the cells using light, so you can assign some causality. So bear in mind, we've made the beta cells all more mature, or at least to the same maturity level. They have more insulin, and they have unanimously the same PDX1 and MAFA levels. So you would expect that islet to secrete more insulin, and you'd expect that islet to be more responsive to glucose, but it's not. We see marked impairments in terms of glucose responses, in terms of ionic fluxes, as well as ability to generate ADP-ADP ratios, which is obviously one of the pivotal steps in stimulus secretion coupling in this cell type. We also see that we lose what we call connectivity, or is just a posh word for coordination. And we see that beta cells normally, you'll see here in the B-norm islets at very fast rates, they kind of flash like lights on a Christmas tree, and are reasonably in sync. But when we kind of lose our immature beta cells, first thing we noticed is we lose hub cells. So we sort of confirmed our original findings on hubs. These are bona fide immature cells. And we also now have an islet where the responses are much more stochastic, because of course we had hypothesized and shown that the hubs are driving coordinated activity in the islets, although the mechanisms of that are somewhat debated. So if we look at insulin secretion, which I guess is the main task of the beta cell, despite having much more insulin content, we have, again, impairments across the board. And interestingly, we had impairments in incretin-stimulated insulin secretion, which we tracked back to lower expression of GLP-1 receptor, which was actually really interesting because GLP-1 receptor is a mature beta cell marker. We've made everything more mature, but at the level of the islet, we're seeing a downregulation. I think this plays into the fact that when you're looking at the transcriptome of cells, it's quite different in cells which are intact within the islet versus those that are dissociated. We're able to recapitulate all this data in human islets. This is using GCAMP6, a genetically encoded calcium indicator. We've now made the human islets have more mature beta cells, and we see again impairments across the board. And we see the reasons for this are due to lowering of voltage-dependent calcium channel expression and function, and we saw the same in rodent islets as well. So this maps quite nicely between species. So what we wanted to know is, okay, we have these cells in the islet, and we knew that when we break apart the islet and we culture the cells in 2 dimensions on the cover slip and we leave them 6 hours, we lose the immature cells. They revert to becoming a PDX1 high, MAFA high state, so more mature. So we wondered, actually, is it the signaling cassette in the islet that's essentially dictating the phenotype of the beta cells throughout the islet? So what we did is we made a chemogenetic model. This is the inch1 Cree, and we expressed H4MDI, which is an inhibitory dread, throughout the beta cell population. You see we get really nice recombination efficiency with this model. And what this dread does is when you put in this designer ligand, CNO or J60, you couple to GI and you reduce cyclic AMP, which essentially reduces electrical activity. And it works really well. These are just control experiments. This is a normal islet. We put in 2nd generation dread ligand here, and we don't see any changes in electrical activity. Whereas in our islet, where we now express the dread, you put J60 in, and within about a minute or so, we get complete silencing. Now, we don't want to silence the islet. We know if we do that, we're going to dedifferentiate everything, because calcium is an important transcriptional signal via mFAT and calcineurin signaling. So we used the 1st generation dread ligand CNO. We dialed the concentration in such that we had about an 8-fold reduction in calcium oscillation frequency. And when we did this, we saw that in our dread islets, we lost our immature cells, both in terms of PDX1 and MAFA. And again, recapitulating the results in our overexpression models, we see, again, defects across the board. Here, I'm showing calcium responses to glucose. So it appears that the immature beta cells are dependent on the signaling in the islet to some extent for their phenotype. So I think what this shows is that the islet is a lot more complex than we think, although not unexpectedly so. I think that heterogeneity is normal for robustness and plasticity. It's unlikely that mechanisms have evolved to constrain this heterogeneity. It's likely to be an advantage. And it's likely going forward that we need approaches where we can actually study, for example, the transcriptome of these cells directly in the intact tissue. And, a big caveat here is we're in an isolated islet, we're still not in the intact pancreas, and we're still not in vivo. And I expect things might change as well there. So I'm just going to switch gears a little bit and now talk about heterogeneity of single proteins on the beta cell itself. And this is work that was spearheaded by another talented postdoc in the lab, Yulia, and was done in collaboration with JB, a close colleague and a very good friend who passed through as a visiting PhD student in the lab a number of years ago when we were doing some photopharmacology stuff with chemistry, and runs his own group now and probably will soon be my boss because he's doing very well indeed. And with JB and Yulia, what we did is we wanted to look at GLP-1 receptors, but we knew that the antibodies suck. And there's been a lot of problems with the antibodies historically. And the antibodies actually weren't very good for what we wanted to do, which was to map the single molecule organization of the receptor on the beta cell surface. So we devised a probe which we termed the luxendins, which is exendin 4,939, so a full antagonist. We mutated the serine position 39 to a cysteine, and then using maleimide file chemistry, we were able to install quite simply a number of different fluorophores, so TMR, which is red, Cy5, which is blinking and good for storm, is far red, and silicon rhodamine, which is fluorogenic and good for STED nonoscopy. And they perform really well. The potency of the luxendins is identical, if not a little bit better than native exendin 9 when we look at cyclic AMP responses. And they open up a whole raft of new experiments that you can do. So this is an islet where we just applied luxendin at 100 nanomolar, left it for an hour, washed it a couple of times. This is a live islet, and then we use 2-photon imaging. And going from top to the bottom, you'll see we've managed to label all the GLP-1 receptors throughout the entire islet. And we've distributed these probes now to about 25 different labs who are using them for various purposes, who are all reporting similar results. So they're a really good tool. And we, at the moment, distributing these via the lab, although it's getting a bit much for us. They will soon be available to buy as well. But of course, we're always happy to collaborate on this. So we wanted to make sure these work well. We, I should say, probably by accident, created a GLP-1 receptor knockout, and you'll see why later. This is single nucleotide deletion, leads to a frame shift of exon 2 and loss of the receptor. And with our luxendin, our GLP-1 receptor antibody, we get nice overlap, and we don't see anything in the knockout animals. So these are really nice and specific. Importantly for us, they open up single molecule imaging experiments. So now we can look at the organization of GLP-1 receptor on the cell surface, and you'll see it's not randomly arranged. It is forming hotspots and clusters on the cell surface, and you have some beta cells which have much higher levels. You can see one here and one here. We don't know what these do yet. We're working on this, but just by looking in high detail, you're able to sort of open up these new experimental paradigms. And this is just the statistical analyses of that, the F function and G function. And we see that about 20% of cells will have very high levels. Working with Davide Calibiro's lab, we did some single molecule imaging just to show the motion of the receptor at the membrane. These are single GLP-1 receptors now labeled, which are flying around the membrane in their unstimulated state. And you will see they're not motionless at all. And in fact, you can break them down into 4 different patterns: so mobile, subdiffusion, normal diffusion, and super diffusion. So subdiffusion is kind of during lockdown when I can't go to the pub. Super diffusion is the moment the pubs reopen in the UK and it hits 4 o'clock and I'm straight out the door going sitting having a pint. And this just shows again the complexity and the detail of the heterogeneity, even the single proteins in terms of the motion heterogeneous. They also work well for sorting beta cells from stem cells. We're able to purify them based upon GLP-1 receptor expression. This is work by Francis Lin, Shugo Sasaki, who we collaborated with. You put on your luxendin onto the beta cell like cells, and you then isolate your luxendin positive fraction. We see they're enriched for GLP-1 receptor. beta cell transcription factors, insulin, and they have less glucagon. And they allow you to look at the brain as well, which is obviously an important site of GLP-1 receptor signaling. This was work with Stefan Trapp and Frank Ryman, and we saw beautiful stain in the arcuate nucleus, the areopostrema, choroid plexus, and again, we see this beautiful organization at single molecule level. So once again, these tools can be used to understand how GLP-1 receptor agonists can access the brain when given via different routes. So we've now taken this one step further. We've made a new suite of Luxendins with different colors with Elisa and Amelia Linneman at Indiana. Again, they work really well. This is just going from sort of green through to near infrared here. Again, they label the islets really well. Lovely and specific. And in particular, Luxendin 758 now allows you to look at beta cell mass and GLP-1 receptor binding in live animals anesthetized using intravital fluorometry approaches. We see nice accumulation of Luxendin 758 in our animals following injection in the sites that you expect. So one of the big things for us is that Luxendins are great. They open up a lot of experimental opportunities and they're really cool to play around with, but they do bind to the GLP-1 receptor and block the orthosteric site, right? So you can't look at things like trafficking and recycling of the receptor. So what we wanted to do is produce mice that had a GLP-1 receptor that has a SNAP tag, an enzyme self-label, and which you can apply a fluorophore which binds virus benzyl guanine moiety to the SNAP. It's covalent, so ergo permanent. You can reverse it if you want to, so that opens up more things, but importantly, it doesn't interfere with binding of orthosteric or allosteric ligands at the GLP-1 receptor. And these animals work really well. You can see this is a new dye we made called sulfode bar, which is membrane impermeable. Again, the dyes are all available for various SNAP labeling and halo labeling functions. And we can label the GLP-1 receptor of the membrane. We can block it first, the SNAP tag, and then we can't label, and we see nothing in the wild types. Again, nice and specific. These are labeled islets from the animals. This is now with Luxendin in the same islet, and you can see beautiful overlay with our orthosteric site bound of a probe and our SNAP label. And the animals are phenotypically completely normal. And they open up new experiments. I mean, this is an experiment that we've taken an intact islet. We're looking at trafficking. Bear in mind that most of our knowledge about GLP-1 receptor trafficking is necessarily performed in immortalized cells, which are heterologously transfected with, for example, your GPCR of choice. But that's very different to what happens in the tissue. I'm sure John will show you more about this later, but you know, you've got glucagon, you've even got GLP-1 in the injured pancreas, and you know, things are going to be slightly different. And these are some very preliminary experiments. We're trying to kind of really dial down on this, but you'll see about 8 minutes following stimulation of our labeled islets, we see some trafficking into the cytosol, and this is reached by 30 minutes, quite profound trafficking of the receptor. And what we see is it's not just every receptor and every cell responds homogeneously, right? What we see is you have subpopulations of cells that are recruited into trafficking. So you have subpopulation number 1, which are early recruited into trafficking, subpopulation 2, which are a little bit later, and subpopulation 3, which are quite late on. So this happens in a very graded manner. And we're trying to understand if this differs between ligands and if this differs with dual agonists as well. So it's a bit of a blast through everything, but hopefully what I've convinced you is that islets are pretty complex, and if you kind of come at these problems a bit naively, I think, as we've done in the past, then you can really dig out new biology. What that means, I don't quite know yet. That's for us in the kind of field to figure out. But I've always been passionate about imaging. I'm a big sort of fanboy of microscopes and everything to do with them. And, you know, the more you can see, the better, right? I mean, if you're going to watch like a sports match in 4K, you're going to see more than if you watch it on an old cathode ray tube TV from the 1970s. And this is the sort of incremental differences we can make with this technology. So the current view is all the cells do the same, all the receptors do the same, and they express all the same surface proteins. However, we're slowly, together with others, updating this view to say that actually the cells don't quite do the same. They have very different traits. They have cell states. Even the receptors on each cell do different things. and heterogeneously organized, and the cells maybe have different roles in the islets as well. So there's loads of, I hope, early career researchers here watching the presentations. I'm actually not far from being early career. I'm very much mid-career at the moment. But what I thought I would do is just warn you about the sort of how much research ages you, right? So this is me at 25 years old when I was going for my postdoc in France. This is me kind of 4 years into a postdoc. I had to learn French as well, which was a nightmare. I had to deal with French administration, which was an even bigger nightmare. So I'm getting grey, but I've got a good tan because I was in Montpellier in the south of France. This was me last year, 38 years old. I was actually 39 in February, and my mum said, I'm going to get you some plastic surgery for your birthday because me and your dad are really worried about your accelerated aging, right? So we went along to the plastic surgeon, and the plastic surgeon said, you know, we can do some stuff on him, and, you know, we can try and sort of halt this, but essentially the best that we can do is this, right? I mean, Keith Richards is pretty much immortal, so I'm happy with that. So a huge amount of people to thank. I've named a lot of them as I've gone along, and certainly put the pictures up. I'm particularly grateful to a lot of awesome people that supply human islets and collaborate with us on this. Very grateful to the funders, but most importantly, the lab. Everybody's shown here, Daniela, Fedi, and the rest designed this little logo, but they called us the Hod Squad. I sort of go along with that, but if it wasn't for them, then we wouldn't be able to do the work we do. And obviously happy to take any questions.